Welcome to G Fun Facts Online. Today, we are diving deep into the architecture of your reality. Imagine for a moment that you are walking into your office. You see your boss sitting at the head of the boardroom table. Your brain instantly retrieves a very specific set of behavioral protocols. You sit up straighter, you speak professionally, you prepare to discuss quarterly targets. The context is professional, and your behavior aligns with that. Now, take that same person, your boss, and imagine seeing them at a heavy metal concert later that night. They are wearing a band t-shirt, they are sweating, and they are moshing in the pit. The object, which is your boss, is identical. The visual data entering your eyes regarding their face and identity has not changed. But the location, the venue, is different. Consequently, the meaning of the event has shifted entirely. Your brain must instantly decouple the identity of the person from the professional context and reattach it to a social, recreational one. For decades, this flexibility was a massive mystery in neuroscience. How does the brain store the concept of boss so that it is stable enough to be recognized anywhere, yet flexible enough to function in completely different situations? The answer, according to a groundbreaking wave of recent research peaking between 2024 and 2025, lies in a specialized architecture of separation. We have finally found what scientists are calling the context neurons. In this episode, we are going to explore the deep, anatomical, and computational mechanisms by which the human brain separates the what, meaning identity, from the where, meaning location, and the which, meaning context. We will look at how the brain miraculously weaves them back together to create the tapestry of episodic memory. To understand the sophisticated machinery of context neurons, we must first look at the foundational highways of the visual brain. The separation of information begins the moment light hits the retina, but it becomes dogmatic once signals reach the primary visual cortex, or V1, at the back of the brain. In 1982, neuropsychologists Leslie Ungerleider and Mortimer Mishkin proposed a theory that would dominate neuroscience for decades called the two-stream hypothesis. Through lesion studies in macaques, they observed that damage to different parts of the brain resulted in two distinct types of blindness. One pathway traveled ventrally, or downward, from the occipital lobe into the temporal lobe. Monkeys with damage here could effectively navigate obstacles and reach for things, but they could not distinguish between a triangle and a square. They had lost the ability to identify what an object was. The other pathway traveled dorsally, or upward, into the parietal lobe. Monkeys with damage here could describe a shape perfectly. They knew it was a square, but they could not tell you where it was. They would reach for it and miss. They had lost the representation of where the object existed in space. This bifurcation suggested that the brain treats identity and location as fundamentally different data types. It makes evolutionary sense. To survive, an organism needs to know what a predator is. Is it a lion or a rock? And it needs to know where it is. Is it coming toward me or moving away? The processing requirements for these tasks are distinct. Identity requires invariance. A lion is still a lion, whether it is seen from the front, side, in light, or shadow. Location requires precision. The exact coordinates relative to the viewer's body are required for flight or fight. The journey of the what pathway is a process of increasing complexity. It begins in V1, which detects simple edges and orientations. As information flows to V2 and V4, the brain begins to process color and curvature. By the time signals reach the inferior temporal cortex, neurons have become remarkably specific. In this cortex, we find cells that do not care about spots of light, but rather about complex shapes. This is the home of the famous Jennifer Aniston neuron, also known as concept cells. This was a discovery made in the early 2000s, where single neurons were found to fire only when a patient saw Jennifer Aniston, whether it was a photo, a drawing, or even just her written name. These content neurons are the masters of semantic identity. They embody the abstract concept of an object. Crucially, they tend to be position invariant. A neuron coding for a banana will fire whether the banana is on the left or right side of your vision. It captures the essence of the object, stripping away the spatial context. Meanwhile, the dorsal stream heads up to the posterior parietal cortex. This system is obsessed with coordinates. It feeds into the motor cortex, guiding our hands and eyes. Historically, this was called the where pathway, but later researchers refined it to the action pathway. They argued that this stream does not just map space for the sake of a map. It maps space to do things. It transforms visual inputs into motor outputs. However, the rigid separation of these streams posed a problem known as the binding problem. If the banana neuron is firing in the temporal lobe and the kitchen counter coordinates are firing in the parietal lobe, how does the brain know that the banana is on the counter? Why don't we hallucinate the banana floating in the garage? The answer lies deeper in the brain, where these two streams finally reunite. The reunion of what and where takes place in the medial temporal lobe, specifically within the structure surrounding the hippocampus. This is the engine room of memory. For years, textbooks describe the hippocampus as a funnel, where everything mixes together. But recent high-resolution fMRI and single-unit recording studies have revealed that the separation of what and where is maintained right up to the doorstep of memory encoding. The gatekeeper of the hippocampus is the entorhinal cortex, and just like the visual cortex, it is divided. The lateral entorhinal cortex, or LEC, receives its primary inputs from the ventral stream. It is the librarian of items. Neurons in the LEC respond to specific objects, odors, and distinct sensory features. If you place a new toy in a rat's cage, the LEC lights up. If you move the toy to a new spot, the LEC doesn't care. It only cares that the toy is there. It is the reservoir of content. On the other hand, the medial entorhinal cortex, or MEC, receives inputs from the dorsal stream and the parietal cortex. It is the librarian of space. This is the home of the Nobel Prize-winning grid cells. These neurons fire in a perfect hexagonal grid pattern as an organism moves through an environment. They provide an internal GPS, a metric of space that is independent of the objects within it. The MEC tells the brain where you are and the geometry of the room, but it is largely blind to what is in the room. This anatomical setup provides a profound insight into how we build memories. We have the MEC providing a spatio-temporal scaffold, essentially a blank grid of space and time. We have the LEC providing the items to populate that grid. Think of it like a theater stage. The MEC builds the stage, the lighting, and the backdrop. The LEC provides the actors and the props. The hippocampus is the director that places the actors onto the stage to create a scene, which we experience as episodic memory. Until very recently, the assumption was that once information reached the hippocampus, it was bound together inextricably. In rodent studies, researchers often found neurons that had mixed selectivity. A neuron might fire only for cheese when it was in the north corner. If you moved the cheese to the south corner, the neuron went silent. This is a conjunctive representation. The what and where are fused. However, the human brain appears to have evolved a more sophisticated layer of flexibility, a mechanism that allows us to decouple these elements even deeper in the memory hierarchy. In January 2026, a landmark study published by researchers at the University of Bonn fundamentally changed our understanding of this process. They utilized a rare opportunity to record directly from the brains of epilepsy patients who had electrodes implanted for surgical planning. The researchers designed a clever task to tease apart content from context. Patients were shown images of various objects, like a biscuit, a dog, or a car. But before each image, they were given a specific task context, represented by a question. Question 1 was, is it bigger? Referring to the object's size being larger than a shoebox. Question 2 was, is it heavier? Referring to the object's weight. This created a matrix of experience. The patient might see a biscuit in the context of the bigger question, or a biscuit in the context of the heavier question. If the brain simply bound these memories immediately, one would expect to find neurons that fire only for the specific combination of biscuit and bigger. The results were startlingly clean. The researchers didn't find a soup of mixed neurons. They found two distinct, segregated armies of cells. First, there were content neurons. These cells fired whenever the biscuit appeared, regardless of whether the question was about size or weight. They represented the object identity in its pure form. Second, there were context neurons. These cells fired whenever the bigger task was active, regardless of whether the image was a biscuit or a dog. They represented the abstract cognitive framework of the task. Unlike rodents, where these representations are often muddied, the human medial temporal lobe maintained a strict division of labor. The most fascinating discovery was how these two groups interacted. They didn't just fire in parallel, they communicated. The researchers observed a temporal delay. The content neurons would fire first, identifying the object, and then, milliseconds later, the context neurons would adjust their firing patterns. It appeared that the object identity was being routed through the context. The authors likened this to a neural library. By storing content and context in separate libraries, the brain achieves infinite combinatorics. If we had to have a separate neuron for every possible combination of object and context, like grandma at the beach, grandma at the store, and grandma in a car, we would run out of neurons. By separating them, the brain can simply link the grandma neuron with the beach neuron on the fly. This is the secret to the flexibility of human memory. It allows us to generalize. We know that grandma is the same person regardless of where she is. Why has the human brain evolved to keep these streams separate for so long? The answer lies in the competing demands of pattern separation and pattern completion. To remember where you parked your car today, you need to distinguish it from where you parked yesterday. The what, which is your car, is identical. The context, which is the space and time, is different. If your brain merged these two early, you would suffer from catastrophic interference. You would walk to yesterday's spot. The dentate gyrus of the hippocampus is famous for this. It takes the inputs from the LEC and MEC and orthogonalizes them. It forces them apart into distinct neural patterns. This ensures that similar experiences don't overwrite each other. Conversely, if you see your car partially obscured by a truck, you need to recognize it as the same car. This requires the what pathway to be robust against noise. By keeping the what representation stable while the where representation shifts, the brain allows you to maintain a stable sense of reality despite a constantly changing environment. How does the brain synchronize these separate libraries? The answer is in the timing. Neuroscientists have long observed theta oscillations, which are 4 to 8 Hz brainwaves, in the hippocampus during exploration. Recent models suggest a phase precession mechanism. Content information might ride on the trough of the theta wave. Context information might ride on the peak of the theta wave. Fast gamma bursts then zip between them, binding the two distinct packets of information into a cohesive conscious moment. This allows the brain to multiplex, transmitting identity and location on the same wire without mixing the signals, simply by using different time slots. The clinical importance of this what versus where separation becomes heartbreakingly clear when we look at diseases that disrupt it. Alzheimer's disease does not attack the brain randomly. It almost always begins in the lateral entorhinal cortex, the gateway of what and content. This explains the specific early symptoms. Patients don't just forget where they are. They often lose the ability to bind objects to contexts. They might recognize a face but have zero idea why they know this person or where they met them. As the disease spreads to the MEC and hippocampus, the map crumbles. The scaffold collapses. The patient becomes lost in their own neighborhood because the grid cells that provide the spatial context can no longer integrate with the visual landmarks. We also see this in Capgras delusion. In this rare psychiatric disorder, a patient recognizes a loved one's face, meaning the what is correct, but believes they are an imposter. Some theories suggest this is a disconnection between the visual what stream and the emotional context provided by the amygdala. The content neuron fires, saying, "That looks like dad," but the emotional context neuron remains silent. The brain resolves this conflict with a delusion, thinking that it looks like dad, but it doesn't feel like dad, so it must be a robot or an alien. Schizophrenia is also often described as a failure of context processing. A healthy brain uses context to inhibit irrelevant associations. If you are at a funeral, you inhibit the urge to laugh. In schizophrenia, the context neurons in the prefrontal cortex and hippocampus may fail to fire or fail to gate the content neurons. The result is a flood of inappropriate associations. A random object, like a red car, feels intensely significant because the brain has failed to place it in its proper neutral context. One of the most exciting developments in the 2025 research landscape is the realization that the where pathway is not just about physical space. It is about cognitive space. The same grid cells that map a room also appear to map conceptual space. In a study where participants had to navigate a space of bird neck lengths and leg lengths, essentially morphing birds on a screen, the MEC fired in a grid pattern. The brain treats relationships as geometry. The bird is the content. The abstract variables of longer neck or shorter legs are the context. This suggests that the context neurons discovered in Bonn are part of a universal machinery. Whether the context is physical location, a task rule, or social hierarchy, the brain uses the dorsal machinery to build a scaffold and hangs the ventral items upon it. This biological separation of what and where is currently inspiring a revolution in artificial intelligence. Traditional convolutional neural networks often struggle with what is called out-of-distribution generalization. If you train an AI to recognize a cow on green grass and then show it a cow on a sandy beach, it might fail. It has overfit the context. It has bound cow and grass into a single, muddy representation. The human brain's architecture, separating content neurons from context neurons, solves this. Newer AI architectures, such as transformers, are attempting to mimic this. They use attention mechanisms to dynamically bind separate representations. They have a query for what they are looking for, representing context. They have a key for what features are available, representing content. By explicitly separating the positional encoding from the token embedding, transformers like GPT-4 function eerily, similarly to the entorhinal cortex. They treat the meaning of a word as separate from its position in the sentence. However, biological brains are still vastly more efficient. A human needs only one example to learn that a smartphone functions the same way in a bedroom as in a boardroom. An eye often needs thousands. The secret likely lies in the physical separation of these neuronal libraries, allowing for instant, zero-shot recomposition of reality. As we move through our lives, we feel a sense of seamless unity. We do not feel the what pathway struggling to identify the coffee cup, while the where pathway calculates the distance to our lips. We do not feel the context neurons switching libraries when we walk from the office to the bar. But this seamlessness is an illusion created by the precise, rhythmic coordination of billions of separated components. The discovery of specialized context neurons in the human brain is the final piece of the puzzle that began with Ungerleider and Mishkin. We now know that the brain is a master of modularity. It deconstructs the world into its atomic parts, identity, location, time, rule, and stores them in separate vaults. It is only in the fleeting electrical present, in the gamma bursts of the hippocampus, that these vaults open, and the scattered notes of reality are bound together into the music of a memory. This architecture of separation is what gives us our freedom. Because our what is not shackled to our where, we can imagine a castle in the sky. We can remember a lost love, without being in the room where we last saw them. We can extract wisdom from one context and apply it to another. In separating the components of our world, the brain gives us the power to transcend them. Thanks for listening to G Fun Facts Online.